In this video we will discuss about efficiency of NR4A2 IL-2 towards promoting host anti-tumor immunity. In the previous video we have understood about the specificity of this uh, tumor specific transgene promoter, it is NR4A2 with IL-2. So in this video they wanted to check whether it can work towards promoting host anti-tumor immunity or not. So they did the studies, in vivo studies in the mice model. So before going into the in vivo studies, they did some ex vivo studies also in the OT1 T cells, where they compare the expressions of the different cytokines, and they compare it with MOC and NR4A2 interleukin 12, and they are able to see that this NR4A2 they are able to see high expressions of this IFN-gamma, TNF, and even the Ki67 markers. And these things they have validated using the, and these things they have also shown the representative image of the flow cytometer, where IFN-gamma and TNF are high in the case where both NR4A2 is being CRISPR knocked in with interleukin 12, and thereafter uh, they quantified these results and shows the reproducibility. So for the case of INF gamma TNF positive and this INF gamma TNF positive, that is the same T cells has both this INF gamma and TNF positives, the same OT1 cells, indicating that these could be the results only because of this NR4A2 with interleukin 12 activations. This Ki67 is also shown that it is also high in the case where this NR4A2 and interleukin 12 is used. And finally they also check which is Tim3, which is the indications for the activated T cells, and this results also in line with the other results that these values are high. There is PD1 positive, Tim3 positives are high in the case where the NR4A2 interleukin 12 is used. So once getting the positive outcomes from the in vitro studies, they did the similar studies in the mice model. First they did the studies with host ova specific CAR T cells, or the mice that has these ovarian cancers, and they use the CAR T construct against this ovarian cancer specific CAR T cell against this ovarian cancer specific antigen along with this NR4A2 and interleukin 12, and they are able to see that the more CD8 plus T cells are found in the spleen, and these things they have shown up in the panel F also, where we can see that in the case of this NR4A2 and interleukin 12, more amount of INF gamma positive and TNF alpha positive cells are present. The quantifications are shown here that further validate their reproducibility. And thereafter they check the studies in the murine CAR T models, where they use the CAR T construct against the murine cells, and they are able to see the same results, that is CD8 plus cells are high in the tumors compared to the other conditions, that is compared to the MOC and NR4A2 knockdown. And even for this proliferation markers is high for this NR4A2 interleukin 12, and the immunofluorescence image also shows the same thing, that is CD8 plus T cells are high in the case where NR4A2 interleukin 12 is used. Then they check it for the spleen, and in the spleen also these values are high for the activated T cells, and also for this IFN gamma and TNF plus positive T cells. So this indicates that, and thereafter, in order to find out why there is a continuous increase in the CD8 plus T cells near the spleen for both the cases, they did the T cell receptor sequencing, and they are able to find out that in the case where NR4A2 interleukin 12s are there, there is more, apart from the proliferations of the cells because of this tetramer that is specific to the antigens that the car is designed, there is also more proliferations of these T cell receptors or the endogenous T cell receptors. There is more proliferations of the endogenous T cell receptors are found in these conditions, that is NR4A2 interleukin 12, and these things they have quantified, or reproducible results are shown in this panel M, or triplicates, results in triplicates from the panel M, where they can see that the more amounts of this endogenous T cell receptors in the case of NR4A2 interleukin 12s are formed compared to the NR4A2 knockdown or mock. So this indicates that their efficiencies of this NR4A2 interleukin 12s is or the NR4A2 interleukin 12 promotes tumor host anti-tumor immunity, not just for the antigens this car is designed, but it will also promote the endogenous T cell receptors enhancement at the site of the tumors, or at the site where there is a, or at the site near the spleen, which is one of the secondary conditions, secondary sites where tumors can form. So that's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.